10 million people around the world are currently living with Parkinson's disease. Most of them are managing symptoms with therapies that, at best, slow things down. There is no cure. And for a long time, researchers focused almost entirely on what was happening in the brain, specifically in the dopamine-producing cells that the disease gradually destroys. But a growing body of research is now pointing somewhere else entirely as a key piece of the Parkinson's puzzle. Not the brain, the gut. And a study out of Nagoya University in Japan has identified a specific chain of events connecting gut bacteria to Parkinson's disease in a way that suggests a treatment so simple it is almost hard to believe. That treatment is B vitamins. Today I want to walk you through exactly what the researchers found, how the chain of events works from your gut all the way to your brain, and what this means for how we might think about Parkinson's in the future. Before I get into the research, I need to give you some context about Parkinson's disease itself, because understanding what the disease does to the brain is essential for understanding why the gut connection matters so much. Parkinson's is a neurodegenerative disease, meaning it progressively destroys nerve cells over time. The cells it targets most directly are the dopamine-producing cells in a region of the brain called the substantia nigra. Dopamine is the neurotransmitter responsible for smooth, controlled movement. When those cells die, movement becomes trembling, rigid, and eventually very difficult to control. But here is something most people do not know. The first symptoms of Parkinson's disease are not motor symptoms at all. They are constipation and sleep problems, and they can appear up to 20 years before the tremors and muscle control loss begin. That detail becomes very important once you understand the gut connection. Scientists have suspected for some time that the relationship between the gut and the brain plays a role in the onset of Parkinson's disease. Previous research had already found that people with Parkinson's experience changes in their gut microbiome long before any of the more recognizable symptoms appear. But the new research from Nagoya University goes further by identifying which specific gut microbes are likely involved and connecting them to a very specific nutritional deficiency. The research team, led by medical researcher Hiroshi Nishiwaki, analyzed fecal samples from 94 patients with Parkinson's disease and 73 relatively healthy controls in Japan. They then compared their results with data from 4 other countries: China, Taiwan, Germany, and the United States. The geographic spread here matters because it means whatever they found was not a quirk of one population's diet or environment. It showed up across completely different countries with very different eating habits and lifestyles. What they found was this. While different groups of bacteria were involved in the different countries examined, they all influenced the same biological pathways, specifically the pathways that synthesize B vitamins inside the body. And in people with Parkinson's disease, those pathways were consistently associated with decreased levels of 2 specific B vitamins: riboflavin, which is vitamin B2, and biotin, which is vitamin B7. Now here's where the chain of events gets important, because this is not just a correlation between low B vitamins and Parkinson's. The researchers mapped out a biological mechanism that explains how the deficiency leads to the kind of damage seen in Parkinson's patients. When gut bacteria fail to adequately synthesize riboflavin and biotin, the body produces fewer short-chain fatty acids and fewer polyamines. These are molecules that play a critical role in maintaining the mucus layer that lines the intestines. That mucus layer is your gut's protective barrier. When it thins out, the intestinal wall becomes more permeable, meaning things that should stay inside the gut can leak through into surrounding tissue. This is sometimes referred to as increased intestinal permeability, and it has been directly observed in people with Parkinson's disease. Once that protective barrier weakens, the intestinal nervous system is exposed to more toxins from the environment. The researchers specifically point to cleaning chemicals, pesticides, and herbicides as examples of the kinds of environmental toxins we now encounter more regularly than previous generations did. These toxins trigger the overproduction of a protein called alpha-synuclein. Alpha-synuclein fibrils are molecules already known to accumulate in the dopamine-producing cells of the substantia nigra in Parkinson's patients. They also drive increased inflammation in the nervous system. Over time, this accumulation and inflammation is what leads to the motor symptoms and dementia that define the later stages of the disease. So the chain, traced backwards from the brain to its origin, looks like this. Environmental toxins enter through a weakened gut lining. The gut lining is weakened because protective molecules are depleted. Those molecules are depleted because B vitamin synthesis in the gut is insufficient. And B vitamin synthesis is insufficient because the gut bacteria responsible for that synthesis have been disrupted. That is a pathway that starts in the gut, runs through the intestinal wall, and ends in the brain. And it potentially begins decades before any brain symptoms appear, which is exactly consistent with the observation that constipation and sleep problems precede Parkinson's motor symptoms by up to 20 years. The treatment implication that follows from this is the part the researchers describe as unexpectedly simple. If the deficiency in riboflavin and biotin is a key driver of the chain of events leading to Parkinson's, then supplementing those B vitamins could potentially interrupt that chain. Nishiwaki and his team propose that identifying individuals with specific deficiencies through gut microbiota analysis or fecal metabolite analysis, and then administering oral riboflavin and biotin supplements to those with decreased levels, could potentially create an effective treatment. In Nishiwaki's own words, supplementation therapy targeting riboflavin and biotin holds promise as a potential therapeutic avenue for alleviating Parkinson's symptoms and slowing disease progression. There is also earlier research supporting this direction. A study from 2003 found that high doses of riboflavin can assist in recovering some motor function in Parkinson's patients, particularly those who also eliminated red meat from their diets. That study is over 2 decades old, which makes the fact that it aligns with this newer mechanistic research all the more interesting. It is important to be clear about what this research does and does not mean. The researchers themselves acknowledge that Parkinson's disease involves a complicated chain of events and that not all patients likely experience the same causes. Some people may have this specific gut bacteria disruption as a major factor. Others may not. Each individual would need to be assessed through gut microbiota or metabolite analysis to determine whether B vitamin supplementation would be relevant for their specific situation. This is not a claim that taking a B vitamin supplement prevents Parkinson's in the general population. It is a finding that a specific biological pathway linking gut bacteria to B vitamin deficiency to intestinal permeability to brain damage appears to be a real and significant part of the picture for at least a meaningful subset of Parkinson's patients. The broader context here is also worth understanding. Researchers are continuously discovering new ways that the composition of our gut bacteria affects our health in areas far beyond digestion. The makeup of the gut microbiome is not fixed. It changes depending on what you eat, how old you are, and even the quality of your sleep. Scientists have linked gut bacteria to insomnia, to autism and ADHD, and most recently have found that certain gut bacteria can absorb and store toxic forever chemicals, potentially offering a way to reduce their harmful effects on the body. The gut is increasingly understood not as a simple digestive organ, but as a major regulatory system with direct influence over the brain, the immune system, and long-term neurological health. For Parkinson's specifically, this research adds to the growing case that what is happening in the gut in the decades before diagnosis is not just a side effect of the disease. It may be part of how the disease gets started in the first place. And if that is true, it opens the possibility of identifying people at risk through gut analysis long before motor symptoms appear and intervening with something as accessible as B vitamin supplementation. The research was published in the journal NPJ Parkinson's Disease. If this kind of science genuinely interests you, hit subscribe and turn on notifications, because I cover research like this every week, explained clearly without the jargon. And share this video with someone who has a family member affected by Parkinson's disease. This is exactly the kind of research they should know about.